Start with the greatest name of Allah Almighty, who is the most merciful and benefit. Assalamu alaikum. Hello, dear student. This is Sikander Ali Sheedi, and so I have request you. So please subscribe and like this YouTube channel. So lecture 1, Phytochrome mechanisms in plant. This topic is assigned by Dr. Abdulaziz Ujan. I have thankful to Sir for giving me a wonderful opportunity to describe here. So this is my contents, history and discovery, introduction to phytochrome, distribution of phytochrome, definition of phytochrome, form of phytochrome, characteristic of phytochromes, mechanisms of phytochromes in plant, role of phytochromes, founding of phytochromes, and references. First of all, history and discovery. The first important work in this field was conducted in 1930s by L.H. and E.D. M.S. Lister. In 1935, they reported that the seed of latex and lactis sativa did not germinate in dark but show very good germination when exposed to wavelength from 525 to 700 nanometer in the red region. H.A. Birdswick and S.P. Hendricks and other 1952 of the U.S. Department of Agriculture Plant Industry Station in Blades Valley, Maryland, using large spectrograph confirmed that finding of lead and MC Lister. So first of all, LH and ED is 1930. He was discovered that the lactus sativa did not germination in dark period, but it is germination in light period. First of all, the wavelength of light is 525 to 700 nanometer is the red region. This point is known as germination to grow for lactus sativa. This is a seed to grow germination in this point. So introduction to phytochrome. Phytochrome is a photoreceptor, a pigment that plant and some algae and fungi use to detect light. It is a sensitive to light in the red and infrared light region of the visible spectrum. Biochemically, phytochrome is a protein white, a billion chromophore. The pigment that control growth and flowering in many plants. In other words, a blue-green pigment found in many plants in which it regulates various developmental processes. So first of all, this phytochrome is also known as a photoreceptor. This is a pigment that is plant some algae and fungi they use to detect light. It is sensitive to touch the plant of the light. They are proper growth of this plant is grow. So distribution of phytochromes. The presence of phytochromes have been known in angiosperm, gymnosperm, liverworts, mosses, ferns, and some green algae. Distribution of phytochrome in grass shoot invariable, but oats, rye, and barley seedling all have high concentration in apical region of the ciliopatile near the shoot apex and in the growing leaf bases. Phytochrome is present in most organs of all plant. Investigating include roots in green lower plant's phytochrome is permanently localized in conformation in or near the plasma membrane. So the definition of phytochrome is a photoreceptor, a pigment that plant and some bacteria and fungi use to detect light. It is sensitive to light in the red and far red region of the visible spectrum. The pigment that controls growth and flowering in many plants, they are almost ubiquitous in green plants, and proteinaceous pigment exist in two forms, PR and PFR form. So this is known as interconvertible changes, is also known as biological activity. So this is a diagram. Phytochrome is a pigment with two interconvertible. The two form of elicit different response. So PR, it means a red light, 670 nanometer, and PFR 730 nanometer for red light. So PR, ये जो पीरियड है यानी इसके अंदर जो लाइट है उसका कोई भी जो है वो रेस्पॉन्स नहीं है, यानी ये 607 नैनोमीटर 70 नैनोमीटर मीन्स रेड लाइट इसको बोलते हैं, और PF और जो होती है इसके अंदर जो है वो फार रेड लाइट मीन्स डार्कनेस पीरियड का हम जिक्र करते हैं. So be sure you understand the difference between red and far red light and two different form of phytochrome. Be sure to understand which form of phytochrome is present when red and far red light on. Form of phytochromes. Phytochromes have two different chemical structures that are interconvertible. The form are named by the color of light that they absorb maximally. PR is a blue form that absorbs red light, and PFR is a blue-green form that absorbs far red light. So PR जो पीरियड है ये एक्टिव इनएक्टिव स्टेट कहलाता है, और PFR जो होता है वो एक्टिव स्टेट कहलाता है. ये दोनों इंटरकन्वर्टेबल चेंजेस है. सो फॉर्म PR form absorb at the peak of 660 nanometer is in form synthesized in dark growing seedling. When PR absorbs red light, it is converted to the PFR form. PFR form absorbed at the peak of 730 nanometer. The PFR is an active form that initiated biological response. When PFR is absorbed for red light, it is converted to the PR form. This form is known as interconvertible changes. कभी कभार ये PR फॉर्म जो होता है वो चेंज हो जाता है PFR के अंदर, और जो PFR फॉर्म होता है वो चेंज हो जाता है PR फॉर्म के अंदर. तो ये इसके अंदर इंटरकन्वर्टेबल चेंजेस होती है. इस इंटरकन्वर्टेबल चेंजेस को हम बायोलॉजिकल एक्टिविटी कहते हैं. कैरेक्टरिस्टिक ऑफ फाइटोक्रोम. The action spectrum of the light needed for these responses show a peak in the red at about 660 nanometer. These responses can be reversed by an application of for light soon after the red treatment. Sensitive spectrophotometers can measure a decrease in plant tissue or exposed to red light. So the changes in absence in caused by the conversion of the photoreceptor from one structural form to another. The red absorbing light The ray absorbing form changes to the far red absorbing form when it absorbs red light and back again when it far red light. The phytochrome molecule is the photoreceptor for red light response. Role of phytochrome. Many flowering plants use it to regulate the time of flowering based on the length of day and night, photoperiodism, and a set of circadian rhythms. It also regulates other response including the germination of seed, photoblast, elongation of seedling, the size, shape and number of leaves, the synthesis of chlorophyll and the stretching of the epicotyle and hypocotyle hook of dicot seedling. It is found in the leaves of most plants. Founding of phytochromes. Phytochromes have been found in most plants including higher plants. Very similar molecules have been found in several bacteria. A fragment of a bacterial phytochromes now has a solid three-dimensional protein structure. So this is a diagram. Sunlight on red PR buildup मीन्स जो रेड लाइट है ये PR को बिल्डअप करती है और जो डार्कनेस जिनी स्लो होती है वो फार रेड लाइट होती है. ये जो होती है वो PFR उसको बिल्डअप करती है. ये जो PFR होती है वो पीरियड जो कहलाता है लॉन्ग डे प्लांट और PR की साइड पे जो पीरियड होता है वो शॉर्ट डे प्लांट होता है. और इसके अंदर फ्लोरोजन एक्टिव दोनों के अंदर होते हैं और दोनों ही क्या करते हैं प्लांट. लेकिन एक जो होता है वो सनलाइट यानी रेड लाइट को कैप्चर करता है और दूसरा जो होता है वो डार्कनेस स्लो यानी फार रेड लाइट को कैप्चर करता है. फाइटोक्रोम मैकेनिज्म्स इन प्लांट वर्क of K.W. Thaman and co-workers suggested that the phytochrome action involved the synthesis of nucleic acid and enzymes which may be responsible for the physiological changes. Moore and others have found the evidence to suggest that phytochrome effects are mediated by activation of genes that code for specific messenger RNA molecules, which is their transcode for a specific enzyme. There are certain responses due to the red and far red light, which are too fast to be explained by the gene activation or repression hypothesis. So this is my references. Thank you for watching my slides. Assalamu alaikum.